Welcome to Science Lecture Site, Medicine Lecture Notes Explained. In today's video, we'll be talking about cell biology lecture notes. I will expect to cover course outline, as you can see, an overview of the cellular basis of life. We'll talk about the structures that make up the cell, the plasma membrane, and transport across the cell membrane. We'll talk about the cytoplasm, cellular organelles, talk about the nucleus, cell division and the cell cycle, and then the diversity of cells. So cell biology is the study of the cellular structure and function. Cells are primarily defined as living structural and functional units enclosed in a membrane. All cells arise from existing cells by the process called cell division, in which one cell divides into two identical cells. Different types of cells fulfill unique roles that support homeostasis and contribute to the many functional capacities of the human organism, as we shall see in our course structure. So, where do cells come from? In the late 1600s, a scientist known as Robert Hooke was looking through a crude microscope at some plant tissue, cork in particular. He saw some cube-like structures that reminded him of the long rows of the monks' rooms in the monasteries. Those rooms are called as cells. So he termed these structures that he saw, he called them as cells. Um, the living cells that had formed that cork were long dead, of course, because cork is made up of dead cells. Only the plant cell walls had remained. However, the name cells has stuck and still being used to describe the smallest unit of living things. Since that time, since the late 1800s, cell research has been exceptionally fruitful and has provided us with the following four concepts collectively known as the cells theory. One, the cell is the basic structure and functional unit of living organisms. So when you define cell properties, you're in fact defining the properties of life. The cell is life. Two, the second statement of the cell theory, the activity of an organism depends on the collective activities of its cells. Three, according to the principle of complementarity, the activities of cells are dictated by their structure, that is, the anatomy of the cell, which determines the functions of the cell, that's the physiology. We'll look at those. And the continuity of life has a cellular basis. In other words, cells come from cells. All existing cells arise from pre-existing cells. We will expand on all these concepts as we get along in our course. Let's begin with the idea that the cell is the smallest living unit. So although one type of cell is exactly like all others, cells do have some basic parts and there are certain functions common to all cells. So briefly, we'll talk about the generalized cell, which demonstrates most of these typical features. In general, all cells have three main regions or parts: a plasma membrane, a nucleus, and the cytoplasm. The nucleus is usually located at the center of the cell, shall see. It is surrounded by the semi-fluid cytoplasm, which in turn is enclosed by the plasma membrane, which forms the outer cell boundary. So for ease of study, we divide the cell into three main parts, as we talked about: the plasma membrane, the cytoplasm, and the nucleus. The cytoplasm is a compartment that has two components, the cytosol and organelles. Each type of organelle has a characteristic shape and specific functions. So examples of organelles include the cytoskeleton, the ribosomes, the endoplasmic reticulum, the Golgi complex, the lysosomes, the peroxisomes, and mitochondria. Within the nucleus, each chromosome, a single molecule of DNA associated with several proteins, contains thousands of hereditary units called genes that control most aspects of cellular structure and function. So let's now look at the plasma membrane. The plasma membrane is a flexible barrier that surrounds and contains the cytoplasm of a cell. The plasma membrane is best described by using the structure model called the fluid mosaic model. According to this model, the molecular arrangement of the plasma membrane resembles an ever-moving sea of fluid lipids that contains a mosaic of many different proteins. Some proteins float freely like icebergs in the lipid sea, whereas others are anchored at specific locations like boats and a dock. We will see the picture of the fluid mosaic model in our next slide. Membranes are fluid structures because the lipids and many of the proteins are free to rotate and move sideways in their own half of the bilayer. The basic structure framework of the plasma membrane is the lipid bilayer. The lipid bilayer simply refers to two lipid layers made up of three types of lipid molecules, the phospholipids, cholesterol, and glycolipids. The bilayer arrangement occurs because the lipids are aliphatic molecules. This means that they have both polar and nonpolar parts. In phospholipids, the polar part is the phosphate-containing head, which is hydrophilic. The nonpolar parts are the two long fatty acid tails, which are hydrophobic hydrocarbon chains. Now, because light seeks light, the phospholipid molecules orient themselves in the bilayer with the hydrophilic heads facing outward. In this way, the heads face a watery fluid on either side, the cytosol on the inside and the extracellular fluid on the outside. The hydrophobic fatty acid tails in each half of the bilayer point toward one another, forming a non-polar hydrophobic region in the membrane's interior. Cholesterol molecules are weakly and fragile. The tiny hydroxide group is the only polar region of the cholesterol, and it forms hydrogen bonds with the polar heads of the phospholipids and the glycolipids. The stiff steroid rings and the hydrocarbon tail of cholesterol are nonpolar. They fit among the fatty acid tails of the phospholipids and glycolipids. The carbohydrate groups of glycolipids form a polar head. Their fatty acid tails are nonpolar. Membrane proteins are classified as integral or peripheral according to whether they are firmly embedded in the membrane. Most integral proteins are transmembrane proteins, which means that they span the entire lipid bilayer and protrude into both the cytosol and extracellular fluids, as you can see in the picture. Like membrane lipids, integral membrane proteins are aliphatic. Their hydrophilic regions protrude into either the watery extracellular fluid or the cytosol, and their hydrophobic regions extend among the fatty acid tails in the lipid bilayer. As the name implies, peripheral proteins are not as firmly embedded in the membrane. They associate more loosely with the polar heads of the membrane proteins, the membrane lipids rather, or with integral proteins at the inner or outer surface of the membrane. Many membrane proteins are glycolipids, are glycoproteins. These are proteins with carbohydrate groups attached to the ends that protrude into the extracellular fluid. The carbohydrates, these proteins are oligosaccharides. Oligosaccharides are made up of few saccharide sugars, chains of 2 to 6 monosaccharides, which may be straight or branched. The carbohydrate portions of glycolipids and glycoproteins form an extensive sugary coat called the glycocalyx. The pattern of carbohydrates in the glycocalyx varies from one cell to another. Thus, the glycocalyx acts as a molecular signature which enables cells to recognize one another. For example, white blood cells' ability to detect a foreign glycocalyx is one basis for the immune response which helps us destroy invading organisms. In addition, the glycocalyx enables cells to adhere to one another in some tissues and protects cells from being digested by enzymes in the extracellular fluid. The hydrophilic properties of the glycocalyx attract a film of fluid to the surface of many cells. This action makes the red cells slippery as they flow through narrow blood vessels and protects cells that line the airways and the gastrointestinal tract from drying out. Those are some of the functions of the glycocalyx, the carbohydrates that are attached to the proteins and lipids of the plasma membrane. So the functions of membrane proteins largely reflect the functions the cell can perform. Some integral membrane proteins form ion channels. Ion channels are holes through which specific ions, such as potassium ions, can flow to get into or out of the cell. Most ion channels are selective, meaning they allow only a single type of ion to pass through. Also, other integral proteins act as carriers. These selectively move a polar substance or ion from one side of the membrane to the other. Carriers are also known as transporters. A carrier transports specific substances across the membrane by changing shape. For example, amino acids needed to synthesize new proteins enter body cells via carriers. Integral proteins called receptors serve as cellular recognition sites. Each type of receptor recognizes and binds to a specific type of molecule. For example, insulin receptors bind the hormone insulin. A specific molecule that binds to a receptor is called a ligand of that receptor. The receptor recognizes a specific ligand and alters the cell's functions in some way. For example, antidiuretic hormone, ADH, binds to receptors in the kidneys and changes the water permeability of certain plasma membranes. Some integral proteins are enzymes that catalyze specific chemical reactions at the inside or outside surface of the cell, depending on which direction the active site faces. An example is lactase protruding from epithelial cells lining the small intestine. This enzyme splits the disaccharide lactose in the milk you drink. Integral proteins may also serve as linkers. These anchor proteins in the plasma membranes of neighboring cells to one another or to protein filaments inside and outside of the cell, thus providing structural stability and shape for the cell. Linkers may also participate in movement of the cell or link two cells together. Membrane glycoproteins and glycolipids often serve as cell identity markers. They enable the cell to recognize other cells of the same kind during tissue formation, or to recognize and respond to potentially dangerous foreign cells. The ABO blood type markers are one example of cell identity markers, which is why when you receive a blood transfusion, the blood type must be compatible with your own. Glycoprotein cell identity markers distinguish your cells from anyone else's. An important class of such markers are the major histocompatibility proteins, the MCH. That's outside the scope of this topic, but it's something you can read about. Peripheral proteins help support the plasma membrane. They anchor integral proteins and participate in mechanical activities such as moving materials and organelles within cells, changing cell shape, and dividing and also cells, and attaching cells to one another. Peripheral proteins also serve as enzymes and linkers. A. Membrane permeability. The term permeable means that a structure permits the passage of substances through it. Impermeable means that a structure does not permit the passage of substances through it. The permeability of the plasma membrane to different substances varies. Plasma membranes permit some substances to pass more readily than others. This property is termed selective permeability. The lipid bilayer portion of the membrane is permeable to non-polar and charged molecules. These include oxygen, carbon dioxide, and steroids. But it is impermeable to ions and large and charged molecules such as glucose. It is also slightly permeable to small and charged polar molecules such as water and urea. Urea is a waste product from the breakdown of amino acids. The slight permeability to water and urea is an unexpected property since they are polar molecules. Transmembrane proteins that act as channels carriers increase the plasma membrane's permeability to a variety of ions and uncharged polar molecules that, unlike water and urea, molecules cannot cross the lipid bilayer unassisted. Channels and carriers are very selective. Each one helps a specific molecule or ion to cross the membrane. Macromolecules, such as proteins, are so large that they are unable to pass across the membrane except by endocytosis and exocytosis processes, which we shall discuss later. So the overview of the plasma membrane now brings us to transport across the cell membrane. Transport of materials across the plasma membrane is essential to the life of a cell. Certain substances must move into the cell to support metabolic reactions. Other substances that have been produced by the cell for export or as cellular waste products must move out of the cell. Substances generally move across cell membranes via transport processes that can be classified as passive or active, depending on whether they require cellular energy. In passive processes, the substance moves down its concentration or electrical gradient to cross the membrane using only its kinetic energy, the energy of motion. Kinetic energy is intrinsic to the particles that are moving. There is no input of energy from the cell. An example of a passive process is simple diffusion. In active processes, cellular energy is used to drive the substance uphill against its concentration of electrical gradient. The cellular energy used is usually in the form of ATP. An example of an active process is active transport. Another way that some substances may enter and leave cells is an active process in which tiny spherical membrane sacs, referred to as vesicles, are used. Examples of such ways of transport include endocytosis, in which vesicles detach from the plasma membrane while bringing materials into a cell, and exocytosis, the merging of vesicles with the plasma membrane to release materials from the cell. Learning why materials diffuse across membranes requires an understanding of how diffusion occurs in a solution. Diffusion is a passive process in which the random mixing of particles in a solution occurs because of the particles' kinetic energy. Both the solutes, the undissolved substances, and the solvent, the liquid that does the dissolving, undergo diffusion. If a particular solute is present in high concentration in one area of a solution and in low concentration in another area, solute molecules will diffuse toward the area of lower solute concentration. The solute molecules move down their concentration gradient. After some time, the particles become evenly distributed throughout the solution, and the solution is said to be at equilibrium. The particles continue to move about randomly due to their kinetic energy, but their concentration does not change at equilibrium. For example, when you place a crystal of dye in a water-filled container, as shown in the figure, the color is most intense in the area closest to the dye because its concentration is higher there. At increasing distances, the color is lighter and lighter because the dye concentration is low. Sometime later, the solution of water and dye will have a uniform color because the dye molecules and water molecules have diffused down their concentration gradients until they are evenly mixed in solution. We now say they are at equilibrium. At the beginning of our experiment, a crystal of dye placed in a cylinder of water dissolves in part M, and then diffuses from the region of higher dye concentration to regions of lower dye concentration in part B. At equilibrium in part C, the dye concentration is uniform throughout, although random movement continues. In this simple example, no membrane was involved. Substances may also diffuse through a membrane if the membrane is permeable to them. However, several factors influence the diffusion rate of substances across membranes. One such factor is the steepness of the concentration gradient. The greater the difference in concentration between the two sides of the membrane, the higher the rate of diffusion. When charged particles are diffusing, the steepness of the electrochemical gradient determines the diffusion rate across the membrane. Another factor that influences the diffusion rate across the membrane is temperature. The higher the temperature, the faster the rate of diffusion. All of the body's diffusion processes occur more rapidly in a person with a fever. The mass of the diffusing substance. The larger the mass of the diffusing particle, the slower its diffusion rate. Smaller molecules diffuse more rapidly than larger ones. Diffusion is also affected by surface area. The larger the membrane surface area available for diffusion, the faster the diffusion rate. For example, the air sacs of our lungs have a large surface area available for the diffusion of oxygen from the air into the blood. This is particularly important as some lung diseases such as emphysema can reduce the surface area. This slows the rate of oxygen diffusion and makes breathing more difficult. Diffusion across a membrane is also affected by the diffusion distance. The greater the distance over which the diffusion must occur, the longer it takes. Diffusion across a membrane takes only a fraction of a second because the membrane is so thin. In diseases like pneumonia, fluid collects in the lungs. This additional fluid increases the diffusion distance because oxygen must move through both the built-up fluid and the membrane to reach the bloodstream. So an increased diffusion distance slows down the rate of diffusion. Um, so now that we have a basic understanding of the nature of diffusion, we will consider the three types of diffusion: simple diffusion, facilitated diffusion, and osmosis. The figure in our slide shows us simple diffusion and the two types of facilitated diffusion, as we shall look at them. In simple diffusion, a substance moves across the lipid bilayer of the plasma membrane without the help of transport proteins. Non-polar hydrophobic molecules move across the lipid bilayer through the process of simple diffusion. Such molecules, which cross the membrane by simple diffusion, include oxygen, carbon dioxide, and nitrogen gases, fatty acids, steroids, and the fat-soluble vitamins A, D, E, and K. Small uncharged polar molecules, such as water, urea, and small alcohols, also pass through the lipid bilayer in simple diffusion. Simple diffusion through the lipid bilayer is important in the movement of oxygen and carbon dioxide between the blood and body cells and between the blood and air within the lungs during breathing. It is also the route of absorption of some nutrients and excretion of some wastes by body cells. Another type of diffusion, precipitated diffusion, solids that are too polar or highly charged to move through the lipid bilayer by simple diffusion can cross the membrane by a passive process called facilitated diffusion. In this process, an integral membrane assists a specific substance across the membrane. The integral membrane protein can be either a membrane channel or a carrier. In channel-mediated facilitated diffusion, A solute moves down its concentration gradient across the lipid bilayer through a membrane channel. Most membrane channels are ion channels. Ion channels are integral transmembrane proteins that allow passage of small inorganic ions that are too hydrophilic to penetrate the nonpolar interior of the lipid bilayer. Each ion can diffuse across the membrane only at certain sites. In typical plasma membranes, the most numerous ion channels are selective for potassium ions or chloride ions. Fewer channels are also available for sodium ions and calcium ions. The diffusion of ions through channels is generally slower than free diffusion through the lipid bilayer because the channels occupy a small fraction of the membrane surface area than the lipids do. However, facilitated diffusion through channels is a very fast process. More than 1 million potassium ions, for example, can flow through the potassium ion channel in 1 second. The channel is said to be gated when part of the channel protein acts as a plug or a gate that changes shape in one way to open the pore and in another way to close it. An example of such a gated channel is the potassium ion channel shown in the figure on our current slide. Some gated channels randomly alternate between the open and closed positions. Others are regulated by chemical or electrical changes inside and outside the cell. When the gates of a channel are open, ions diffuse into or out of cells down the electrochemical gradients. The plasma membranes of different types of cell may have different numbers of ion channels and thus may display different permeability to various ions. In another type of facilitated diffusion, carrier-mediated facilitated diffusion, a carrier, also called as a transporter, is used to move a solute down its concentration gradient across the plasma membrane. As you can see in the figure on our slide, the solute binds to a specific carrier on one side of the membrane and is released on the other side after the carrier undergoes a change in shape. The solute binds more often to the carrier on the side of the membrane with a higher concentration of solute. Once the concentration is the same on both sides of the membrane, solute molecules bind to the carrier on the systolic side and move out to the extracellular fluid as rapidly as they bind to the carrier on the extracellular side and move into the cell. The rate of carrier-mediated facilitated diffusion, that is how quickly this diffusion occurs, is determined by the steepness of the concentration gradient across the membrane. The number of carriers available in the plasma membrane places an upper limit, also called as a transport maximum, on the rate at which facilitated diffusion can occur. Once all of the carriers are occupied, the transport maximum is reached, and a further increase in the concentration gradient does not increase the rate of facilitated diffusion. So carrier-mediated facilitated diffusion exhibits a kind of saturation. Substances that move across the membrane by carrier-mediated facilitated diffusion include glucose, fructose, galactose, and some vitamins. Glucose enters many body cells by carrier-mediated facilitated diffusion, as shown in this figure here. Glucose binds, as you can see, glucose binds to a specific type carrier protein called the glucose transporter on the outside surface of the membrane. As the transporter undergoes a change in shape, glucose passes through the membrane. The transporter releases glucose on the other side of the membrane, the inside. The selective permeability of the plasma membrane is often regulated uh, with the aim to achieve homeostasis. For example, the hormone insulin, which acts via insulin receptor molecules, promotes the insertion of many copies of glucose transporters into the plasma membrane of certain cells. Thus, the effect of insulin is to elevate the transport maximum, facilitating the diffusion of glucose into cells. With more glucose transporters available, body cells can pick up glucose from the blood more rapidly. An inability to produce or utilize insulin is a medical condition called as diabetes mellitus. The third example of the extrusion, the third type of the extrusion is osmosis. Osmosis is a type of the extrusion in which there is net movement of the solvent through selectively permeable membrane. Like the other types of the extrusion, osmosis is a passive process. It doesn't require energy. In living systems, the solvent is water. This water moves by osmosis across plasma membranes from an area of higher water concentration to an area of lower water concentration. In terms of solute concentration, in osmosis, water moves through a selectively permeable membrane from an area of lower solute concentration to an area of higher solute concentration. During osmosis, water molecules pass through the plasma membrane in two ways. By moving through the lipid bilayer via a simple diffusion, or they move through the bilayer through aquapolines. Aquapolines are integral membrane proteins which function as water channels. Osmosis occurs only when a membrane is permeable to water, but is not permeable to certain solids. So a simple experiment can help demonstrate this principle of osmosis. Consider a U-shaped tube in which a selectively permeable membrane separates the left and right arms of the tube. A volume of water is poured into the left arm, and the same volume of solution containing a solute that cannot pass through the membrane is poured into the right arm. Because the water concentration is higher on the left and lower on the right, net movement of water molecules, that is osmosis, occurs from left to right, so that the water is moving down its concentration gradient. At the same time, the membrane prevents the diffusion of the solute from the right arm into the left arm, as the membrane is selectively permeable. As a result, the volume of water in the left arm decreases, and the volume of solution in the right arm increases. So you might think that osmosis would continue until no water remained on the left side, but this is not what happens. In this experiment, the higher the volume of solution in the right arm becomes, the more pressure it exerts on each side of the membrane. The pressure exerted in this way by liquid, also known as hydrostatic pressure, forces water molecules to move back into the left arm. Equilibrium is reached when just as many molecules move from right to left due to hydrostatic pressure as the same number move from left to right due to osmosis. Also, the solution with the impermeable solute also exerts a force called the osmotic pressure. The osmotic pressure of a solution is proportional to the concentration of the solute particles that cannot cross the membrane. So the higher the solute concentration, the higher the solution's osmotic pressure. So now consider what would happen if a piston were used to apply more pressure to the fluid in the right arm of the tube. As you can see in part C of the figure, with enough pressure, the volume of fluid in each arm could be restored to the starting volume, and the concentration of solute in the right arm would be the same as it was at the beginning of the experiment. The amount of pressure needed to restore the starting condition equals the osmotic pressure. So in our experiment, osmotic pressure is the pressure needed to stop the movement of water from the left tube into the right tube. It is important to note that osmotic pressure of a solution does not produce the net movement of water during osmosis. Rather, it is the pressure that will prevent such water movement. Normally, the osmotic pressure of the cytosol is the same as the osmotic pressure of the interstitial fluid outside the cells. Because the osmotic pressure on both sides of the plasma membrane, which is selectively permeable, is the same, cell volume remains constant, relatively. When body cells are placed in a solution having a different osmotic pressure than the cytosol, however, the shape and volume of the cells change. As water moves by osmosis into or out of the cells, their volume increases or decreases. As you can see, the solution's tonicity is a measure of the solution's ability to change the volume of cells by altering their water content. Any solution in which a cell, for example, the red cell, maintains its normal shape and volume is an isotonic solution. Isotonic, same, iso. The concentrations of solutes that cannot cross the plasma membrane are the same on both sides of the membrane in this solution. For example, a 0.9% sodium chloride solution, which is 0.9 grams of sodium chloride in 100 milliliters of solution. Such a solution, also called as normal or physiological saline solution, is isotonic for red cells. The red cell plasma membrane permits the water to move back and forth, but it behaves as though it is impermeable to sodium ions and chloride ions, the solutes in this case. Any sodium ions and chloride ions that enter the cell through channels or transporters are immediately moved back out by active transport or by other means. When red cells are bathed in 0.9% sodium chloride, water molecules enter and exit at the same rate, allowing the red cells to keep their normal shape and volume. Cells placed in an isotonic solution maintain their shape because there is no net water movement into or out of the cell. Now, a different situation results if the red cells are placed in a hypotonic solution, a solution that has a lower concentration of solutes than the cytosol inside the red cells. In this case, water molecules enter the cells faster than they leave, causing the red cells to swell and eventually to burst. The rupture of red cells in this manner is called hemolysis. The rupture of other types of cells due to placement in a hypotonic solution is simply referred to as lysis. Pure water is very hypotonic. It causes rapid hemolysis in red cells and lysis in other cells. A hypertonic solution, hyper, greater than, has a higher concentration of solutes than does the cytosol inside the red cells. One example of a hypertonic solution is 2% sodium chloride solution. In such a solution, water molecules move out of the cells faster than they enter, causing the cells to shrink. Such shrinkage of cells is called crenation. So as we've seen, red cells and other body cells may be damaged or destroyed if exposed to hypertonic or hypotonic solutions. For this reason, most intravenous or IV solutions, these are liquids infused into the blood of the vein, are isotonic. IV solutions, most of them are isotonic. Examples of such isotonic solutions include isotonic saline, we talked about that, 0.9% sodium chloride, and dextrose 5% in water. Sometimes infusion of a hypertonic solution, such as mannitol, is useful to treat patients who have a condition like cerebral edema, which is excessive interstitial fluid in the brain. Infusion of such a hypertonic solution relieves fluid overload by causing osmosis of water from interstitial fluid into the blood. Then the kidneys excrete this excess water from the blood into the urine. Hypotonic solutions, given either orally or through an IV, can be used to treat people who are dehydrated. The water in the hypotonic solution moves from the blood into the interstitial fluid, then into the body cells to hydrate them. The water and most sports drinks that you consume to hydrate after a workout are actually hypotonic relative to your body cells. So, having looked at the fusion and its three types, we now look at the active processes of transport across the membrane, starting with active transport. Now, remember that there are some polar or charged solutes that must enter only body cells and yet cannot cross the plasma membrane through any form of passive transport because they would need to move uphill against their concentration gradients. Such solutes cross the membrane by a process called active transport. Active transport is considered an active process because energy is required for the carrier proteins to move the solutes across the membrane against their concentration gradient. Two sources of cellular energy can be used to drive active transport. The energy obtained from hydrolysis of adenosine triphosphate, which is ATP, is the source in primary active transport. Energy stored in an ionic concentration is the source in secondary active transport. Just as carrier-mediated facilitate the diffusion, active transport processes also exhibit a transport maximum and saturation. Solutes actively transported across the membrane include ions such as potassium ions, sodium ions, hydrogen ions, calcium ions, iodide ions, and chloride ions. Amino acids and monosaccharides are also transported actively across the membrane. Note that some of these substances also cross the membrane via facilitated diffusion when the proper channel proteins or carriers are present, as we described under facilitated diffusion. So having talked about the two types of active transport, primary and secondary, we now look at the first of the two, primary active transport. In primary active transport, energy derived from the hydrolysis of ATP changes the shape of a carrier protein, which pumps the substance across a plasma membrane against its concentration gradient. As a result, by the way, carrier proteins that mediate primary active transport, often called pumps. A typical body cell spends about 40% of the ATP it generates on primary active transport. And chemicals that turn off ATP production, for example, the poison cyanide, are very dangerous because they shut down active transport in cells throughout the body. The most prevalent primary active transport mechanism expels sodium ions from cells and brings potassium ions in. Because of the specific ions it moves, this carrier is referred to as the sodium-potassium pump. Because a part of the sodium-potassium pump acts as an ATPase, the enzyme that hydrolyzes ATP. Another name for this pump is the sodium ion potassium ion ATPase. All cells have thousands of sodium potassium pumps in their plasma membrane. These sodium potassium pumps maintain a low concentration of sodium ions in the cytosol by pumping them into the extracellular fluid against the sodium ion concentration. At the same time, these pumps move potassium ions into the cells against the potassium ion concentration gradients. Because sodium ions and potassium ions slowly leak back across the plasma membrane down the electrochemical gradients, basically through passive transport or secondary active transport, the sodium-potassium pumps must work non-stop to maintain a low concentration of sodium ion and a high concentration of potassium ion in the cytosol. The figure on this slide depicts the operation of the sodium-potassium pump. 3 sodium ions in the cytosol bind to the pump protein. Binding of the sodium ions triggers the hydrolysis of ATP into ADP. This reaction also attaches a phosphate group to the pump protein. This chemical reaction changes the shape of the pump protein, expelling the 3 sodium ions into the extracellular fluid. Now the shape of the pump protein favors binding of 2 potassium ions in the extracellular fluid to the pump protein. The binding of potassium ions triggers release of the phosphate group from the pump protein. This reaction again causes the shape of the pump protein to change. As the protein pump reverts to its original shape, it releases potassium ions into the cytosol. And at this point, the pump is again ready to bind 3 sodium ions and the cycle repeats. The different concentrations of sodium ions and potassium ions in cytosol and extracellular fluids are crucial for maintaining normal cell volume and for the ability of some cells to generate electrical signals, such as action potentials. By helping to maintain normal tonicity on each side of the plasma membrane, the sodium-potassium pumps ensure that cells neither shrink nor swell due to the movement of water by osmosis out into the cells. Second type of active transport is secondary active transport. Secondary active transport mechanisms use the energy stored in an ionic concentration gradient to drive other substances across the membrane against their own concentration gradients. Because a sodium ion or hydrogen ion gradient is established by primary active transport pumps that hydrolyze ATP, secondary active transport indirectly uses energy from the hydrolysis of ATP. The sodium-potassium pump maintains a steep concentration of sodium ions across the plasma membrane. As a result, these sodium ions have stored potential energy. Just like you would keep water behind a dam, it accumulates energy. Accordingly, there is a route for these sodium ions to leak back into the cell. Some of the stored energy can be converted into kinetic energy, the energy of motion, and can be used to transport other substances against their concentration gradient. As you can see in the figure, in essence, secondary active transport proteins harness the energy in the sodium ion concentration gradient by providing routes for sodium ions to leak into cells. In secondary active transport, a carrier protein simultaneously binds to sodium ion and another substance and then changes its shape so that both substances cross the membrane at the same time. If these transporters move two substances in the same direction, they are called symporters. Antiporters, in contrast, move two substances in opposite directions across the membrane. Plasma membranes contain several antiporters and symporters that are powered by the sodium ion gradient. For example, the concentration of calcium ions is low in the cytosol because the sodium ion, calcium ion antiporters eject calcium ions. This is a very important principle which has a significant application in medicine. A drug called Digitalis is often given patients with heart failure. Heart failure is a condition of weakened pumping action by the heart. This drug exerts its effect by slowing down the action of sodium-potassium pumps, which lets more sodium ions to accumulate inside the heart muscle cells. The result is a decreased sodium ion concentration gradient across the plasma membrane, which causes the sodium ion, calcium ion antiporters to slow down. As a result, more calcium remains inside heart muscle cells, and the increase in the level of calcium ions in the cytosol of heart muscles increases the force of their contractions and thus strengthens also the heartbeat. As we noted earlier, other substances, a variety of them, are transported in vesicles from one structure to another within cells and across the cells. Vesicles also import materials from and release the materials into the extracellular fluid. During endocytosis, which is one type of transport in vesicles, materials move into a cell in a vesicle formed from the plasma membrane. In exocytosis, materials move out of a cell by the fusion with the plasma membrane of vesicles from inside the cell. Both processes, endocytosis and exocytosis, require energy supplied by ATP, and thus are active processes. Endocytosis Under endocytosis, we consider the three types of endocytosis, which are receptor-mediated endocytosis, phagocytosis, and bulk phase endocytosis. Receptor-mediated endocytosis is a highly selective type of endocytosis by which cells take up specific ligands. Remember, we said ligands are molecules that bind to a specific receptor. A vesicle forms after a receptor protein in the plasma membrane recognizes and binds to a particular particle in the extracellular fluid. Uh, Endocytosis is important for uptake of cholesterol-containing low-density lipoproteins, LDLs. Transferrin is an iron transporting protein in the blood, the protein that transports iron. Some vitamins, antibodies, and certain hormones are also taken up by receptor-mediated endocytosis. As you can see in the figure on the slide, receptor-mediated endocytosis of an LDL occurs as shown. On the first step, binding. On the extracellular side of the plasma membrane, a low-density uh, lipoprotein particle that contains cholesterol binds to a specific receptor in the plasma membrane, form a receptor LDL complex. The receptors are integral membrane proteins that are concentrated in regions of the plasma membrane called creatine-coated pits. Here, a protein called creatine attaches to the membrane on its cytoplasmic side. Many creatine molecules come together and form a basket-like structure around the receptor LDL complexes, causing the membrane to fold inward. The process is called as invagination. The folded edges of the plasma membrane around the creatine-coated pit fuse, and a small piece of the membrane pitches off. The resulting vesicle, also called as the creatine-coated vesicle, contains the receptor LDL complexes. In step 3, almost immediately after it is formed, the creatine-coated vesicle loses its creatine coat to become an uncoated vesicle. Creatine molecules either return to the inner surface of the plasma membrane or help form coats on other vesicles inside the cell. In stage 4, fusion with the endosome. The uncoated vesicle quickly fuses with a vesicle known as an endosome. Within the endosome, the LDL particles separate from their receptors. Stage 5, recycling of receptors in plasma membranes. Most of the receptors accumulate in elongated protrusions of the endosome. These pinch off, forming transport vesicles that return the receptors to the plasma membrane. An LDL receptor is returned to the plasma membrane about 10 minutes after it enters the cell. Other transport vesicles which contain the LDL particles bud off the endosome and soon fuse with the lysosome. The lysosomes contain many digestive enzymes. Certain enzymes break down the large protein and lipid molecules of the LDL particle into amino acids, fatty acids, and cholesterol. These smaller molecules then leave the lysosome. The cell uses cholesterol for rebuilding its membranes and for synthesis of steroids such as estrogen. The fatty acids and amino acids can be used for ATP production or to build molecules needed by the cell. Phagocytosis is a form of endocytosis in which the cell engulfs large solid particles, such as worn-out cells or bacteria viruses. Only a few body cells called as phagocytes are able to carry out phagocytosis. Two main types of phagocytes are the macrophages, located in many body tissues, and the neutrophils, a type of white blood cell. Phagocytosis begins when the particle binds to a plasma membrane receptor on the phagocyte, causing it to extend pseudopods. These are projections of the plasma membrane and cytoplasm. Pseudopods surround the particle outside the cell, and the membranes fused form a vesicle called a phagosome. The phagosome enters the cytoplasm and fuses with one or more lysosomes, and in the lysosome, the lysosomal enzymes break down the ingested material. In most cases, undigested materials in the phagosome remain indefinitely in a vesicle called the residual body. The process of phagocytosis is very vital in defense mechanisms that protect the body against disease. Through phagocytosis, macrophages dispose of invading microbes and worn out red cells. Neutrophils also help the body or help rid the body of invading microbes. Uh, pus is a mixture of dead neutrophils, macrophages, and tissue cells and fluid in an infected wound. Most body cells carry out bulk phase endocytosis, also called as pinocytosis, which is a form of endocytosis in which tiny droplets of extracellular fluid are taken up. No receptor proteins are involved. All solutes dissolved in the ECF are brought into the cell. During bulk phase endocytosis, the plasma membrane folds inward and forms a vesicle containing a droplet of the ECF. The vesicle detaches or pinches off from the plasma membrane and enters the cytosol. Within the cell, the vesicle fuses with the lysosome, where the enzymes degrade the engulfed solutes. The resulting smaller molecules leave the lysosome to be used elsewhere in the cell. So, um, um, receptor-mediated endocytosis and phagocytosis are more like a form of cell eating, uh, where we take in more like solid material, and pinocytosis is more of like cell drinking. uh, the cells take up fluids in pinocytosis. Having described the three types of endocytosis, we now turn our attention to exocytosis. In contrast with endocytosis, which brings materials into the cell, exocytosis releases materials from the cell. All cells carry out exocytosis. However, it is especially important in two types of cells: the secretory cells that liberate digestive enzymes, hormones, mucus, and other secretions, and the nerve cells that release substances called neurotransmitters. In some cases, in some cells, wastes are also released by exocytosis. During exocytosis, membrane-enclosed vesicles called secretory vesicles from inside the cell fuse with the plasma membrane and release their contents into the ECF. Segments of the plasma membrane lost through endocytosis are actually recovered or recycled by exocytosis. So this balance between endocytosis and exocytosis keeps the cell surface, the cell's plasma membrane, relatively constant. Membrane exchange is quite extensive in certain cells. In the pancreas, for example, the cells that secrete digestive enzymes can recycle an amount of plasma membrane equal to the cell's entire surface in just about 90 minutes. Another type of transport in vesicles, called as transcytosis, um, involves using transport vesicles to successively move a substance into, across, and out of a cell. Across. In this active process, in transcytosis, vesicles undergo endocytosis on one side of the cell, move across the cell, and then undergo exocytosis on the opposite side rather. As the vesicles fuse with the plasma membrane, the vesicular contents are released into the ECF. Transcytosis occurs most often across the endothelial cells that line blood vessels and is a means for materials to move between the blood plasma and the interstitial fluids. A particular example is when a woman is pregnant. Some of her antibodies cross the placenta into the fetal circulation via transcytosis. So a full account of the transport across the cell membrane now brings us to looking at the cytoplasm and cellular organelles, their structure, composition, and function. The cytoplasm consists of all the cellular contents between the plasma membrane and the nucleus and has two compartments. The cytosol and the organelles. The organelles are the tiny structures that perform different functions in the cell. The cytosol, also called as intracellular fluids, is the fluid portion of the cytoplasm that surrounds the organelles. Constitutes about 55% of total soil volume. Although it varies in composition and consistency from one part of the cell to another, the cytosol is about 75 to 90% water, along with various dissolved and suspended components. Among these are the different types of ions, glucose, amino acids, fatty acids, proteins, lipids, ATP, waste products, all those are suspended in the cytosol. Also present in some cells are various organic molecules that aggregate into masses for storage. These aggregations may appear and disappear at different times in the life of a cell. Examples of such molecules include the lipid droplets that contain triglycerides and clusters of glycogen molecules called as glycogen granules. The cytosol is the site of many chemical reactions required for a cell's existence. For example, the enzymes in the cytosol catalyze glycolysis. These are a series of 10 chemical reactions that produce two molecules of ATP from one molecule, glucose. Other types of cytosolic reactions provide the building blocks for maintenance, cell structures, and for cell growth. The organelles are specialized structures within the cell that have characteristic shapes. They perform specific functions in cellular growth, maintenance, and reproduction. Despite the many chemical reactions going on in a cell at any given time, there is a little interference among reactions because they are confined to different organelles. Each type of organelle has its own set of enzymes that carry out specific reactions and serves as a functional compartment for specific biochemical processes. The numbers and types of organelles vary in different cells depending on the cell's function, as we shall see. Although they have different functions, organelles often cooperate to maintain homeostasis. The nucleus is a large organelle with special importance in directing the life of a cell. We shall discuss the nucleus in a specific section, other than that for the organelles. So looking at the first type of organelle, the cytoskeleton, is a network of protein filaments that extend throughout the cytosol. It serves as a scaffold that helps to determine a cell's shape and to organize cellular contents. It aids movement of organelles within the cell, of chromosomes during cell division, and of whole cells, such as phagocytes. Three types of filamentous proteins contribute to the cytoskeleton structure, as well as the structure of other organelles. In order of the increasing diameter, these structures are the microfilaments, intermediate filaments, and microtubules. The microfilaments are the thinnest elements of the cytoskeleton. They are composed of a protein called actin and are most relevant at the edge of the cell. As you can see in this um, figure, microfilaments are closer to the edge of the cell as compared to the other filaments of the cytoskeleton. Microfilaments have two general functions. They help generate movement and they provide mechanical support. With respect to movement, microfilaments are involved in muscle contraction, cell division, and the cell locomotion, such as occurs during migration of embryonic cells during development. The cell locomotion also occurs during the invasion of tissues by white cells to fight infection, or in the migration of skin cells during wound healing. These microfilaments provide much of the mechanical support that is responsible for the basic growth, for the basic strength rather, and shapes of cells. They anchor the cytoskeleton to integral proteins in the plasma membrane. They also provide mechanical support for cell extensions called microvilli. These are non-motile, microscopic, finger-like projections of the plasma membrane. Within each microvillus is a core of parallel microfilaments that support it. Because they greatly increase the surface area of the cell, microvilli are abundant on cells involved in absorption, such as the epithelial cells that line the small intestine. As their name suggests, intermediate filaments are thicker than microfilaments, but thinner than microtubules. Several different proteins can compose intermediate filaments, which are exceptionally strong. They are found in parts of cells subject to mechanical stress. They help stabilize the position of organelles, such as the nucleus, and they help attach cells to one another. The third type of filaments that make up the cytoskeleton, the microtubules, are the largest of the cytoskeletal components and are long and branched hollow tubes composed mainly of the protein tubulin. The assembly of microtubules begins in an organelle called the centrosome. The microtubules grow outward from the centrosome toward the periphery of the cell. Microtubules also help determine cell shape. They also function in the movement of organelles such as secretory vesicles, movement of chromosomes during cell division, and movement of specialized cell projections such as cilia and flagella. Microtubules are the most dominant components of cilia and flagella. These are the multi-projections of the cell surface. Cilia are numerous, short, hair-like projections that extend from the surface of a cell. Each cilium contains a core of 20 microtubules surrounded by a plasma membrane. The coordinated movement of many cilia on the surface of a cell causes the steady movement of fluid along the cell's surface. Many cells of the respiratory tract, for example, have hundreds of cilia that sweep foreign particles trapped in mucus out from the lungs. This is particularly important as since in conditions like cystic fibrosis, extremely thick mucus secretions that are produced interfere with ciliary action and the normal functions of the respiratory tract. The movement of cilia is also paralyzed by nicotine, a chemical found in cigarette smoke. For this reason, smokers cough often, remove foreign particles from their airways. Also, the cells that line the uterine or the fallopian tubes have cilia which sweep oocytes or egg cells towards the uterus, and females who smoke have an increased risk of an ectopic pregnancy outside the uterus because their cilia may not be able to move fertilized over to the uterus for implantation. Flagella are very similar in structure to cilia but are typically much longer. Flagella usually move an entire cell in forward motion. The only example of a flagellum in the human body is the sperm cell's tail, which propels the sperm toward the oocyte in the uterine tube. Another cellular organelle, the ribosomes, are the sites of protein synthesis. The name of these tiny organelles reflects the high content of ribosomal RNA. Structurally, a ribosome consists of two subunits, one about half the size of the other, as you can see in the figure in the top right corner of our slide. The large and small subunits are made separately in the nucleolus, a spherical body inside the nucleus. Once produced, the large and small subunits exit the nucleus separately and then come together in the cytoplasm. Some ribosomes are attached to the outer surface of the nuclear membrane and an extensively folded membrane called the endoplasmic reticulum. These ribosomes synthesize proteins destined for specific organelles for insertion into the plasma membrane or for export from the cells. Other ribosomes are free or they are not attached to other cytoplasmic structures. Free ribosomes synthesize proteins used in the cytosol. Ribosomes are also located within mitochondria, where they synthesize mitochondrial proteins. The endoplasmic reticulum, another cell organelle, is a network of membranes in the form of flattened sacs or tubules. The endoplasmic reticulum extends from the nuclear envelope at the membrane around the nucleus, which it is connected and spread throughout the cytoplasm. The endoplasmic reticulum is so extensive that it constitutes more than half of the membranous surfaces within the cytoplasm of most cells. Cells contain two distinct forms of endoplasmic reticulum which differ in structure and function. The rough endoplasmic reticulum is continuous with the nuclear membrane and is usually folded into a series of flattened sacs. The outer surface of the rough endoplasmic reticulum is studded with ribosomes. These are the sites of protein synthesis. Proteins synthesized by ribosomes attached to the rough endoplasmic reticulum enter spaces within the ER for processing and sorting. In some cases, enzymes attach the proteins to carbohydrates to form glycoproteins. In other cases, enzymes attach the proteins to phospholipids, also synthesized by the rough endoplasmic reticulum. These molecules may be incorporated into the membranes of organelles, be inserted into the plasma membrane, or may be excreted via exocytosis. Thus, the rough endoplasmic reticulum produces secretory proteins, membrane proteins, and many organelle proteins. The smooth endoplasmic reticulum, the second type of endoplasmic reticulum, extends from the rough endoplasmic reticulum to form a network of membrane tubules. Unlike the rough endoplasmic reticulum, the smooth endoplasmic reticulum does not have ribosomes on the outer surfaces of its membrane. However, smooth ER contains unique enzymes that make it functionally more diverse than the rough endoplasmic reticulum. Because it lacks ribosomes, the smooth endoplasmic reticulum does not synthesize proteins. Rather, it synthesizes fatty acids and steroids such as estrogen and testosterone. In liver cells, enzymes of the smooth endoplasmic reticulum help release glucose into the bloodstream and inactivate or detoxify lipid-soluble drugs or potentially harmful substances such as alcohol, pesticides, and carcinogens. Carcinogens are cancer-causing agents. In the liver, kidney, and intestinal cells, a smooth endoplasmic reticulum enzyme removes the glucose 6-phosphate group, or the phosphate group from glucose 6-phosphate, which allows free glucose to enter the bloodstream. In muscle cells, calcium ions that trigger the contraction of muscle cells are released from the sarcoplasmic reticulum, which is actually a form of smooth endoplasmic reticulum. So most of the proteins synthesized by ribosomes attached to the rough endoplasmic reticulum are ultimately transported to other regions of the cell. The first step in the transport pathway is through another cell organelle called as the Golgi complex. This Golgi complex, also called as the Golgi apparatus, consists of cavities known as cisternae. The cisternae, all these cavities, are often curved, and this gives the Golgi complex a cup-like structure. Although most cells have several Golgi complexes, these complexes are more extensive in cells that secrete proteins, and this is an important clue to the organelle's role in the cell. The cisternae at the opposite ends of the Golgi complex differ from each other in size, shape, and enzymatic activity. The convex entry, or C-space, is a cisterna that faces the rough endoplasmic reticulum. The concave exit, or transface, is a cisterna that faces the plasma membrane. Sacs between the entry and exit faces are called mediocisternae. Transport vesicles from the rough endoplasmic reticulum merge from the entry phase. From the entry phase, the cisternae mature, in turn becoming medial and then exit cisternae. Different enzymes in the entry, medial, and exit cisternae of the Golgi complex permit each of these areas to modify, sort, and package proteins for transport to different destinations. The entry phase receives and modifies proteins produced by the rough ER. The mediocystine add carbohydrates to proteins to form glycoproteins and lipids to proteins to form lipoproteins. The exit phase modifies the molecules further and then sorts and packages them for transport to their destinations. Another cell organelle, the lysosomes, are membrane-enclosed vesicles that form from the Golgi complex. Inside these lysosomes, as many as 60 kinds of powerful digestive and hydrolytic enzymes can break down a wide variety of molecules once the lysosomes fuse with vesicles formed during endocytosis. Because lysosomal enzymes work best at an acidic pH, the lysosomal membrane includes active transport pumps that import hydrogen ions. Thus, the lysosomal interior has a pH of 5. This is about 100 times more acidic than the pH of the cytosol, which is pH 7, chemically neutral. The lysosomal membrane also includes transporters that move the final products of digestion, such as glucose, fatty acids, and amino acids, into the cytosol. Lysosomal enzymes also help recycle worn-out structures. A lysosome can engulf another organelle, digest it, and return the digested components to the cytosol for reuse. In this way, old organelles are continually replaced. The process by which entire worn-out organelles are digested is called autophagy. In autophagy, the organelle to be digested is enclosed in a membrane derived from the endoplasmic reticulum, creates a vesicle called as the autophagosome. The vesicle then fuses with the lysosome. In this way, for example, a human liver cell recycles about half of its cytoplasmic contents every week. Autophagy is also involved in cellular differentiation, lateral growth, and tissue remodeling, adaptation to adverse environments, and cell defense. Lysosomal enzymes may also destroy the entire cell that contains them, a process known as autolysis. Autolysis occurs in some pathological conditions and is also responsible for the tissue deterioration that occurs immediately after death. As we just discussed, most lysosomal enzymes act within a cell. However, some operate in extracellular digestion. One example occurs during fertilization, where the head of a sperm cell releases lysosomal enzymes that aid its penetration of the oocyte by dissolving its protective coating in a process called as the acrosomal reaction. Another group of organelles similar in structure to lysosomes, but smaller, are the peroxisomes. Peroxisomes, also called as microbodies, contain several oxidases. These are enzymes that oxidize or remove hydrogen atoms from various organic substances. For example, amino acids and fatty acids are oxidized in peroxisomes as part of normal metabolism. In addition, enzymes in peroxisomes oxidize toxic substances such as alcohol. Thus, peroxisomes are very abundant in the liver, where detoxification of alcohol and other damaging substances occurs. A byproduct of the oxidation reactions is hydrogen peroxide, a potentially toxic compound. However, peroxisomes also contain the enzyme catalase, which decomposes hydrogen peroxide. So because the production and degradation of the peroxide occur within the same organelle, peroxisomes protect other parts of the cell from the toxic effects of the peroxide. And just like mitochondria, lysosomes can self-replicate, rather peroxisomes can self-replicate. New peroxisomes may form from pre-existing ones by enlarging and dividing. They may also form by a process in which components accumulate at a given site in the cell and then assemble into a peroxisome. Now we know that lysosomes degrade proteins delivered to them in vesicles. Cytosolic proteins also require disposal at certain times in the life of a cell. So this continuous destruction of unneeded, damaged, or faulty proteins is the function of tiny barrel-shaped structures called as proteasomes. A typical body cell contains many thousands of proteasomes in both the cytosol and the nucleus. These proteasomes are so named because they contain proteases, the enzymes that cut proteins into small peptides. Once the enzymes of a proteasome have broken down a protein into smaller molecules, other enzymes then break down the peptides into amino acids, which can be recycled into new proteins. Proteasomes are very important as some diseases could result from their failure to degrade abnormal proteins. For example, clumps of misfolded proteins may accumulate, have been found to accumulate in brain cells of people with Parkinson's and Alzheimer's. Another cell organelle, the mitochondrion, consists of an outer mitochondrial membrane and an inner mitochondrial membrane with a small fluid-filled space between them. Both membranes are similar in structure to the plasma membrane. The inner mitochondrial membrane contains a series of folds called cristae. The central fluid cavity of a mitochondrion, enclosed by the inner mitochondrial membrane, is the mitochondrial matrix. The elaborate folds of the cristae provide an enormous surface area for the chemical reactions that are part of the aerobic phase of cellular respiration. These are the reactions that actually produce most of the cell's ATP. The enzymes that catalyze these reactions are located on the cristae and in the matrix of the mitochondria. Because they generate most of the cell's ATP through aerobic respiration or oxygen requiring respiration, mitochondria are also referred to as the powerhouses of the cell. A cell may have as few as 100 or as many as several thousand mitochondria, depending on its activity. Active cells, such as those found in the muscles, the liver, the kidneys, which use ATP at a high rate, have a large number of mitochondria. Mitochondria are located in cells that use oxygen or those parts of the cell where ATP is used. For example, in lung, the contractile proteins are in muscle cells. Just like peroxisomes, mitochondria self-replicate. This occurs mainly during times of increased cellular activity that demands a lot of energy, or even just before cell replication. Synthesis of some of the proteins needed for mitochondrial functions occurs on the ribosomes that are present in the mitochondrial matrix. Mitochondria even have their own DNA in the form of multiple copies of a circular DNA molecule that contains 37 genes. These mitochondrial genes control the synthesis of two ribosomal RNAs, 22 transfer RNAs, and some other 13 proteins that build mitochondrial compartments. Now, note that although the nucleus of each somatic cell contains genes from both the mother and the father, mitochondrial genes are inherited only from your mother. The head of the sperm, the part that penetrates and fertilizes the oocyte, normally lacks most organelles like the mitochondria, the ribosomes, the plasmic reticulum, the Golgi complex, and any sperm mitochondria that do enter the oocyte are soon destroyed. So that brief overview of the cytoplasm and the organelles um, brings us to yet another important cell organelle, yet a special one, the nucleus. The nucleus is a spherical or oval-shaped structure that usually is the most prominent feature of a cell. Most cells have single nucleus, although some, such as mature red cells, have none. However, some cells, such as the skeletal muscle cells and a few other types of cells, can have multiple nuclei. Um, Talking about the structure of the nucleus, a double membrane called the nuclear envelope separates the nucleus from the cytoplasm. Both layers of the nuclear envelope are lipid bilayers, similar to the plasma membrane. The outer membrane of the nuclear envelope is continuous with the rough endoplasmic reticulum and resembles it in structure. Many openings called nuclear pores extend through the nuclear envelope. Each nuclear pore consists of a circular arrangement of proteins surrounding a large central opening that's about 10 times wider than the pore of a channel protein in the plasma membrane. Nuclear pores control the movement of substances between the nucleus and the cytoplasm. Small molecules and ions move through the pores passively by diffusion. Most large molecules, such as RNAs and proteins, cannot pass through the nuclear pores by diffusion. Instead, their passage involves an active transport process in which the molecules are recognized and selectively transported through the nuclear pore into or out of the nucleus. Um, for example, the proteins needed for nuclear functions move from the cytosol into the nucleus, and in a similar manner, newly formed RNA molecules are moved from the nucleus into the cytosol. Inside the nucleus are one or more spherical bodies called nuclei, and these function in producing ribosomes. Each nucleolus is simply a cluster of protein, DNA, and RNA that's not enclosed by a membrane. Nucleoli are the sites of synthesis of ribosomal RNA and assembly of the ribosomal RNA and proteins into ribosomal units. Nucleoli are quite prominent in cells that synthesize large amounts of protein, such as muscle and liver cells. Nucleoli disperse and disappear during cell division, reorganize once new cells are formed. Within the cell nucleus are most of the cell's hereditary units called genes, which control cellular structure and direct cellular activities. Genes are arranged along chromosomes. The human somatic cells, the body cells of the human beings, have 46 chromosomes, okay, with 23 inherited from each parent. Each chromosome is a long molecule of DNA that is coiled together with several proteins. This complex of DNA, proteins, and some RNA is called chromatin. The total genetic information carried in a cell or an organism is referred to as its genome. So this talk about genes and chromatids, chromosomes, brings us to the next section of our discussion, cell division, the cell cycle. Most cells of the human body undergo cell division, the process by which cells reproduce themselves. The two types of cell division, somatic cell division and reproductive cell division, accomplish different goals for the organism. A somatic cell is any cell of the body other than a germ cell. Human cells contain 23 pairs of chromosomes for a total of 46. One member of each pair is inherited from each parent. The two chromosomes that make up each pair are called homologous chromosomes. These homologs contain similar genes arranged in the same or almost the same order. When examined under a light microscope, homologous chromosomes generally look very similar. The exception to this rule is one pair of chromosomes called as the sex chromosomes, designated as X and Y. In females, the homologous pair of sex chromosomes consists of two large X chromosomes, and in males, the pair consists of an X and a much smaller Y chromosome. Because somatic cells contain two sets of chromosomes, they are called as diploid cells, 2N. When a cell reproduces, it must duplicate all of its chromosomes to pass its genes to the next generation of cells. The germ cell is a gamete, the sperm or the oocyte, or any precursor cell destined to become a gamete. In somatic cell division, the cell undergoes a nuclear division called mitosis and a cytoplasmic division called cytokinesis. Produce two identical cells, each with the same kind of chromosomes as the original cell. Somatic cell division replaces dead or redundant cells and adds new ones during tissue loss. Reproductive cell division is the mechanism that produces gametes, the cells needed to form the next generation of sexually reproducing organisms. Reproductive cell division consists of a special two-step division called meiosis, in which the number of chromosomes in the nucleus is reduced by half. Somatic cell division is best described using the cell cycle model. The cell cycle consists of two major periods. Interphase, when the cell is not dividing, and the mitotic phase, when the cell is dividing. In a complete cell cycle, the cell duplicates its contents and divides into two identical cells. During interphase, the cell replicates its DNA. It also produces additional organelles and cytosolic components in anticipation of cell division. Interphase is a state of high metabolic activity, as it is during this time that the cell does most of its growing. Interphase consists of three phases: G1, S, and G2, as you can see in the figure on our slide. The S stands for synthesis of DNA. So because the G phases are periods where there is no activity related to DNA duplication, they are thought of as gaps or interruptions in DNA duplication. The G1 phase is the interval between the mitotic phase and the S phase. During G1, the cell is metabolically active, replicating most of its organelles and cytosolic components except for its DNA. Replication of centrosomes also begins in the G1 phase. So for a cell with a total cell cycle of 24 hours, G1 lasts about 8 to 10 hours. Note that the duration of this phase is quite variable. It can be very short in embryonic cells or in cancer cells. Cells, there are some that remain in G1 for a long time and perhaps never divide again. These are said to be in G0 phase. Most nerve cells are actually in G0 phase. Once a cell enters the S phase, it is committed to both way of cell division. The S phase is the interval between G1 and G2, can last around 8 hours. During this phase, DNA replication occurs, and the resulting um, uh, uh, two identical cells formed during cell division will have the same genetic material as a result of this DNA replication. The G2 phase is the interval between the S phase and the mitotic phase. It lasts around 5 hours. During the G2 phase, cell growth continues, enzymes and other proteins are synthesized in preparation for cell division, and the replication of centrosomes is completed. So a microscopic view of a cell during interphase will show a clearly defined nuclear envelope, a nucleolus, tangled mass of chromatin, as you can see in step 1 of our figure on this slide. Once a cell completes its activities during G1, S, and G2 phases of interphase, mitotic phase begins. So the mitotic phase of the cell cycle consists of nuclear division and a cytoplasmic division or cytokinesis. Nuclear division is the mitosis. This mitotic phase results in formation of two identical cells. The events that occur during mitosis and cytokinesis are plainly visible under microscope because chromatin condenses into discrete chromosomes. Mitosis is the distribution of two sets of chromosomes into two separate nuclei. This process results in the exotic partitioning of genetic material. For convenience, biologists divide the process of mitosis into four stages: prophase, metaphase, anaphase, and telophase. As you can see on the figure, on step 2, during early prophase, the chromatin fibers condense and shorten into chromosomes that are visible under the light microscope. The condensation process may prevent entangling of the long DNA molecules, DNA strands, as they move during mitosis. Because DNA replication took place during the S phase of interphase, each prophase chromosome consists of a pair of identical strands called chromatids. A constricted region called a centromere holds the chromatid pair together. At the outside of each centromere is a protein complex called as the kinetochore. Later in prophase, tubules in the pericentroidal material of the centrosomes start from the mitotic spindle. The mitotic spindle, a football-shaped assembly of microtubules that attach the kinetochore. As the microtubules lengthen, they push the centromeres to the poles, to the ends of the cell, so that the spindle extends from pole to pole. The mitotic spindle is responsible for the separation of chromatids to opposite poles of the cell. The nucleolus then disappears and the nuclear envelope breaks down. During metaphase, step 3 on our figure, the microtubules of the mitotic spindle align the centromeres of the chromatid pairs at the exact center of the mitotic spindle. This midpoint region is called as the metaphase plate. During mitotic anaphase, step 4, the centromeres split, separating the two members of each chromatid pair, which move toward opposite poles of the cell. Once separated, the chromatids are termed chromosomes. As the chromosomes are pulled by the microtubules of the mitotic spindle during anaphase, they appear V-shaped because the centromeres lead the way, dragging the trailing arms of the chromosomes toward the pole. The final stage of mitosis telophase, stage 5 on the figure, begins after chromosomal movement stops. The identical sets of chromosomes, now at opposite poles of the cell, uncoil and revert to the thread-like chromatin form. A nuclear envelope forms around each chromatin mass. Nuclei reappear in the identical nuclei, and the mitotic spindle breaks up. Following mitotic telophase, division of a cell's cytoplasm and organelles into two identical cells is called cytokinesis. This process usually begins in late anaphase with the formation of a cleavage furrow. This is the slight indentation of the plasma membrane and is completed after telophase. The cleavage furrow usually appears midway between the centrosomes and extends around the periphery of the cell, as you can see in step 5 on our figure. Actin microfilaments that lie just inside the plasma membrane form a contractile ring that pulls the plasma membrane progressively inward. This ring constricts the center of the cell, just like we tie a belt around the waist, and ultimately pitches it into two. Because the plane of the cleavage furrow is always perpendicular to the mitotic spindle, the two sets of chromosomes end up in separate cells. When cytokinesis is complete, interphase begins again. In a process called sexual reproduction, each new organism is a result of the union of two different gametes. Fertilization. Each gamete produced by a different parent. If gametes had the same number of chromosomes as somatic cells, the number of chromosomes would double at fertilization. Meiosis, the reproductive cell division that occurs in the gonads, in the ovaries and testes, produces gametes in which the number of chromosomes is reduced by half. Fertilization restores the diploid number of chromosomes. So unlike, as a result of this meiosis, gametes contain a single set of 23 chromosomes, and that's why they are termed as haploid cells. And fertilization restores the diploid state. Unlike mitosis, which is complete after a single round, meiosis occurs in two successive stages, meiosis 1 and meiosis 2. During interphase, the interphase that precedes meiosis 1, the chromosomes of the diploid cell start to replicate. As a result of this replication, each chromosome consists of two sister or genetically identical chromatids, which are attached at their centromeres. This replication of chromosomes is similar to the one that precedes mitosis and somatic cell division. Meiosis 1, which begins once chromosomal replication is complete, consists of 4 phases: phase 1, metaphase 1, anaphase 1, and telophase 1. So phase 1 is an extended phase in which the chromosomes shorten and thicken. The nuclear envelope and nuclei disappear, the mitotic spindle forms. Two events that are not seen in mitotic prophase occur during phase 1 of meiosis. First, the two sister chromatids of each pair of homologous chromosomes pair up, an event called synapsis. The resulting 4 chromatids form a structure called a tetrad. Second, parts of the chromatids of two homologous chromosomes may be exchanged with one another. Such an exchange between parts of non-sister or genetically different chromatids is termed as crossing over. This process, among others, permits an exchange of genes between chromatids of homologous chromosomes. Due to crossing over, The resulting cells are genetically unlike each other and genetically unlike the starting cell that produced them. Crossing over results in genetic recombination, formation of new combinations of genes, and accounts for part of the genetic variation among humans and other organisms form gametes via meiosis. In metaphase 1, the tetrads formed by the homologous pairs of chromosomes line up along the metaphase plate of the cell with homologous chromosomes side by side. During anaphase 1, the members of each homologous pair of chromosomes separate as they are pulled opposite poles of the cell by the microtubules attached to the centromeres. The paired chromatids held by the centromeres remain together. Recall that during mitotic anaphase, the centromeres split and the sister chromatids separate. Telophase 1 and cytokinesis of meiosis are similar to telophase and cytokinesis of mitosis. The net effect of meiosis 1 is that each resulting cell contains the haploid number of chromosomes because it contains only one member of each pair of the homologous chromosomes present in the starting cell. The second stage of meiosis, meiosis 2, also consists of 4 phases: phase 2, metaphase 2, anaphase 2, and telophase 2. These phases are similar to those that occur during mitosis. Centromeres split, the sister chromatids separate and move towards opposite poles of the cell. So in summary, meiosis 1 begins with a diploid cell and ends with 2 cells, each with the haploid number of chromosomes. During meiosis 2, each of these haploid cells formed during meiosis 1 divides. The net result is 4 haploid gametes that are genetically different from the original diploid starting cell. So that overview of cell division and the cell cycle now brings us to the last section of our discussion on cellular diversity. The body of an average human adult is composed of nearly 100 trillion cells. All these cells can be classified into just about 200 different cell types. Body cells vary greatly and considerably in size. The sizes of cells are measured in units called as micrometers. 1 micrometer is equal to millions of a meter. High-powered microscopes are needed to see the smallest cells of the body. The largest cell in the body, the egg cell, the oocyte, has a diameter of about 140 microns and is just barely even visible to the unaided eye. The largest, the red cell, for example, has a diameter of 8 microns. To better visualize this, know that an average hair from the top of your head is approximately 100 microns in diameter. The shapes of cells also vary considerably. They may be round, oval, flat, cube-shaped, column-shaped, elongated, star-shaped, cylindrical, or disc-shaped. A cell's shape is related to its function in the body. Cellular diversity also permits organization of cells into more complex tissues and organs. The shapes of cells and the relative numbers of the various organelles they contain relate to specialized cell functions. Let's now take a look at some examples of specialized cells. We have cells that connect body parts. The fibroblast is a cell that has an elongated shape, just like the cable-like fibers that it secretes. It has an abundant rock in the plasmic reticulum and the Golgi apparatus to make and secrete the protein building blocks of those fibers. The red blood cell, the erythrocyte, is a cell that carries oxygen in the blood. Its biconcave shape provides extra surface area for the uptake of oxygen and streamlines the cell so it flows easily through the bloodstream. So much oxygen-carrying pigment is packed in erythrocytes so that all other organelles have been ejected to make room. This is a particularly great adaptation of red cells to their oxygen-carrying function in the body. Among cells that cover and line body organs is the epithelial cell. The hexagonal shape of this cell is exactly like a cell in a honeycomb of a beehive. This shape allows the epithelial cells to pack together in sheets, one sheet on top of the other. An epithelial cell has abundant intermediate filaments and desmosomes that resist tearing when the epithelium is rubbed or pulled. Um, Cells that move organs, body parts include the skeletal, cardiac, and smooth muscle cells. These cells are elongated and filled with abundant contractile microfilaments so that they can shorten forcefully and move the bones, pump blood, or change the size of internal organs to move substances around the body. The fat cell is one example of cells that store nutrients. The huge spherical shape of the fat cell is produced by a large lipid droplet in its cytoplasm. White blood cells are cells that fight diseases, including macrophage, phagocytic cell. This cell extends long pseudopods, force feet, to probe through the tissue to reach infection sites. The many lysosomes within the cell digest the infectious microorganisms, such as the bacterium, which it essentially eats. The nerve cell or the neuron is a cell that gathers information and controls body functions. This cell has long processes, extensions, for receiving messages and transmitting them to other structures in the body. The processes of the nerve cell are covered with an extensive plasma membrane and a plentiful rough endoplasmic reticulum synthesizes membrane components and the signaling molecules called as neurotransmitters. The cells of reproduction are the oocyte in the female, the sperm in the male. The oocyte is the largest cell in the body. This egg cell contains several copies of all organelles for distribution to the other cells that arise when the fertilized egg divides to become an embryo. The sperm, the reproductive cell of males, is long and streamlined, built for swimming to the egg for fertilization. Its flagellum acts as a motile whip to propel the sperm. Oh, thank you for coming this far. Thank you for watching this video to the end and for learning a lot of science and medicine with us. Please remember to like, share your comments, and hit the subscribe button for more interesting videos on medicine like channels. Thank you.